Okay, let's start off with our Lipids Concept Map. I'm going to zoom in. We're going to look at the general characteristics associated with our lipids. And our lipids provide 9 calories per gram. And the reason why is because they are highly reduced. Big long hydrocarbon, we can remove a lot of hydrogens and get a lot of energy from that during the oxidative process. Our lipids are nonpolar, and that's because they contain that hydrocarbon, which of course is going to drive the whole reason why we get 9 calories per gram from our lipids compared to our proteins and our carbohydrates. Solid lipids at room temperature are known as your fats, and liquid lipids at room temperature are known as your oils. If we look at our functions of our lipids, that would include storage, structure, and signaling, just like our proteins that we went over for our last exam. So if we start looking at our storage, the simplest of our lipids are called our fatty acids. Now I have a note here, know the tables in the notes, the ones that we were tested over for our fatty acid quiz, but also don't forget to know the sources for this exam as well. With our simple fatty acids, we can break them down as to whether they're saturated or unsaturated. With our saturated fatty acids, those are the ones that have 0 double bonds. No double bonds, they're completely saturated with hydrogens. Our MUFAs and PUFAs are the ones that have some double bonds in there at least. These collectively are referred to as our unsaturated fatty acids. With MUFAs, they just have 1 double bond, and so 1 double bond, and that double bond can either be cis or trans. There are a few trans double bonds found in nature, but that's usually dairy products are the main source. Most of the trans that we consume are unnatural, synthetic ones that are formed via partial hydrogenation. Remember, partial is that key word. When we hydrogenate something, we're taking away double bonds. So if we partially hydrogenate it, we're taking away some of the double bonds, but not all of them. And the ones that are left over are trans compared to when they were in the natural form, they were in cis formation. So if we were to completely hydrogenate the structure, we would take away all of the double bonds. We would end up with a saturated structure. We do nothing to the number of carbons within the molecule of the fatty acid, but we just get rid of all of the double bonds. So with our PUFAs, polyunsaturated fatty acids, those are the ones that have 2 or more double bonds. Those are the essential fatty acids, which of course would be your omega-3s and 6s. And the more double bonds you have, the more likely it is to go rancid. So it's more likely to go bad and have a shorter shelf life. Over here on the left, it says the main form stored in muscle and adipose tissue is going to be your triglycerides, or triacylglycerol is another name for it. The backbone of your triglycerides is the glycerol molecule. And triglycerides are formed via dehydration or condensation. And remember, we're forming ester bonds in there, and that's called esterification. Okay, so if I go back to the left now, that's the end of our storage form of it. Structure's next. With structure, we have our membrane lipids. The membrane lipids could be glycolipids, or if we scooch up here, we have our phospholipids, or the main type. Phospholipids are going to be degraded via phospholipases. Don't forget, make sure you go back and know what phospholipase A1 breaks, A2, C, and D. The backbone of the phospholipids can either be a glycerol or it can be a sphingosine molecule. If it's a glycerol backbone with 2 fatty acids. It's going to be made from the parent compound called phosphatidic acid. The most common ester glycerol phospholipid is lecithin or phosphatidylcholine, or some of them can have ether bonds in them. Remember that carbon, oxygen, carbon, that's an ether bond. Whereas in esters, carbon, oxygen, carbonyl, and then carbon again. So it looks like almost a carboxylic acid stuck between some carbons. We had 2 ether glycerophospholipids, that's plasmalogen and platelet activating factor, which is important for clotting, activates platelets. With the ester glycerophospholipids like lecithin, make sure you go back to the table that's in our notes, know the 5 most common glycerophospholipids, and some of the important concepts related to them, like which ones carry the omega-3s, omega-6s. With our sphingospholipids, that has a sphingosine backbone, there's only one fatty acid attached to our sphingosine backbone, and the only sphingophospholipid that we talked about was sphingomyelin. With our sphingosine is made from a ceramide. Our sphingosine backbone plus a fatty acid makes up a ceramide. If you remember that, ceramides are signaling compounds, but if you have too much, then it can be really inflammatory. Accumulation, the genetic defect that causes accumulation of ceramide is known as Farber disease, and the genetic defect that causes accumulation of sphingomyelin is your Niemann-Pick. If we go back to our glycolipids, the other type of membrane lipids that we have, glyco tells us it's attached to carbohydrates. So these are carbs and lipids together. We have some that are called cerebrosides. Cerebrosides are one sugar. They contain one sugar on them. And that sugar can either be glucose. If it's glucose, it's called a glucocerebroside, and it's located in non-neural tissue. If that sugar is galactose, it's located in neural tissue, and it's called galactocerebrosides. Accumulation of galactocerebrosides will lead to Krabbe disease, and accumulation of glucocerebrosides will lead to Gaucher. It looks like Goucher, but it's Gaucher. Glycolipids that contain a ceramide plus some sugars, 2 to 4 sugars, are called globosides. Nothing super exciting about them. And then ceramide plus, we have 3 plus sugars, so multiple sugars. We're looking at polysaccharides, oligosaccharides there. And N-acetylneurominic acid. Those are your gangliosides. Now the types of sugars that we have in there and the pattern that they're attached in will give you different gangliosides. So we'll have different disorders related to which ones will accumulate. So we have accumulation of one type of ganglioside will lead to Sandoff's disease. Another type of ganglioside that will accumulate will lead to Tay-Sachs. Okay, other membrane lipids include cholesterol. Remember, cholesterol is really important functionality within the membrane itself. Cholesterol is going to be made in the liver. That slowest enzyme, the rate-limiting enzyme in the process of making our endogenous cholesterol, is called HMG, CoA reductase. That's one that statins will inhibit, decrease cholesterol synthesis within the liver. Cholesterol is a precursor for vitamin D3, some of our steroid hormones, including things like estrogen and testosterone. The storage form of cholesterol is known as a cholesterol ester. And we can turn it into bile as well. So bile acids, which is just free cholate. If it's conjugated, they're called bile salts or conjugated bile acids. That would be your glycocholate and taurocolate. Now, as signaling molecules, we have intracellular signaling and extracellular signaling. With our extracellular signaling, the main extracellular are the steroid hormones. So I have a line going from extracellular signaling all the way down to our steroid hormones, which was our estrogen, testosterone, cortisol. Intracellular signaling, though, we have ceramides, phosphatidylinositol, and then our essential fatty acids. Phosphatidylinositol was used in the protein kinase C pathway. Where we had phospholipase C will break off the polar head group and the phosphate to give us IP3, and that releases calcium that acts as an intracellular messenger to, along with diglyceride, will go ahead and activate protein kinase C. You remember that kinases are important for phosphorylation, so all protein kinase C is just going to phosphorylate another protein, which is either going to make it more active or less active. Ceramides we've already talked about. Those are the sphingosine backbone and a fatty acid to make up ceramide. Too much ceramide has been associated with insulin resistance. And a lot of inflammation. And then lastly, we have our eicosanoids that are made from arachidonic acid, or they're made from EPA. If we start off with arachidonic acid's 20 carbons, 4 double bonds, that was your omega-6. It can either go through the COX or the LOX pathway. If it goes through the COX pathway, then we're going to synthesize our 2-series prostaglandins, and we'll also synthesize our 2-series thromboxanes, which are made by our platelets. And the 2-series thromboxanes in particular are important for clotting. The 2-series prostaglandins are really inflammatory also. Remember that COX takes away 2 double bonds, so if we start off with 4 double bonds, COX takes away 2, we're going to end up with 2 double bonds. We get the 2-series prostaglandins and thromboxanes. The cyclooxygenase enzyme is going to be inhibited by aspirin. Our LOX enzyme does not do anything to the number of double bonds, so if you start off with 4, you end up with 4. We get the 4-series leukotrienes, which are associated with asthma, allergy, inflammation, and most of our lipoxygenase is located within our white blood cells. EPA is the other fatty acid that can go through the COX or LOX pathway because it's 20 carbons. 20 carbons, 5 double bonds, it's our omega-3. I said COX takes away 2 double bonds, so if we start off with 5 double bonds, we end up with the 3-series prostaglandins and the 3-series thromboxanes, which really don't do much. If we go through the LOX pathway, start off with 5 double bonds, we end up with the 5-series leukotrienes. Thus, the LOX enzyme can also make our resolutionary compounds, which are protectants and resolvins, which are the anti-inflammatory cleanup mechanism. Now, EPA, of course, is found in high concentration with DHA and a lot of fish. So with this, there are some fish to avoid, like tilefish from the Gulf of Mexico, shark, swordfish, and king mackerel. These are all very large fish that accumulate more mercury, and it's been found that those that consume higher levels of mercury from these fish can actually have a decrease in glutathione concentration, which is an antioxidant. If this happens, you end up with a redox balance, or imbalance basically, so you have a lot more oxidation in the brain, causing some dysfunction. Okay, that is the end of our first lipids concept map. Thank you very much.